share?
Yeah. Do I turn on my microphone?
What's that?
Do I need to turn on the microphone here?
No, it's like in the somewhere in the ethos or something. Ether.
Okay. All right. Hi, my name is Jules. I'm a second-year student in the Krupitzer lab of evolutionary neurobiology. And I'm going to present the organization of cortical patterns in the context of evolution and experience. So all mammals have a partitioned neocortex, and we call these partitions cortical fields. Here you're seeing an evolutionary tree that has images of the cortex across mammals. And you can see the primary visual area is in blue, the primary somatosensory area is in red, and the primary auditory area is in green. And we call these areas primary because they're the first regions to receive inputs from the thalamus that connects directly to the sensory system. So like from the visual system, that's the first inputs from the eyes to the thalamus to the cortex. So it's considered primary visual area. From there, there's visual processing, like higher order visual processing in the secondary areas, if that makes sense. So how do we come to define cortical fields? These were originally defined by Brodmann in 1909. So Brodmann divided the entire expanse of the human cerebral cortex into 52 areas. And he found these areas based on staining. So he was taking tissues and applying Nissl stain and myelin stain. And myelin stains like the fatty tissue around axon terminals. So the myelin stained areas come out darker than the other areas. And this he defined as a region, those darker areas. And then later in the 1900s, it was shown that through electrophysiological recordings that each of these areas is functionally different, and the neurons in those areas respond differently to different features in the environment. So in the Krupitzer lab, what we do is we extract the brains from a variety of different species. We flatten and make slices out of the brains, and then we stain the slices. So here you're seeing, this is cytochrome oxidase staining, and this is myelin staining. So you can see in the myelin stain already, like there's these darker regions that you can clearly just see with your naked eye. But we take the slices and we go into the microscope room, and then we draw the slices, we draw the lines around the blobs of the darker stains for a bunch of different slices, and we kind of get a visual average of where those regions are for a given brain sample. So here you're seeing flattened tissue from a lab rat that's been stained for myelin, and you can kind of already see these regions. And then after we trace these by hand, we then trace them in Illustrator, and we assign a color to each of the regions. So in red, you're seeing a primary somatosensory area, green is primary motor area, blue is primary visual area, purple is primary auditory area, and kind of can't see that, but this, no. Anyway, gray is piriform cortex, which is responsible for olfaction. So once we have this tracing, we can measure the shape of these fields, the total area of the field, and the location of that field on the cortex. And we assign a color to each of these regions, and the colors are totally random. They're just to differentiate between regions. So what I've been doing is tracing these fields in a standardized way across all of the existing, or as many as possible of the existing species that have already been experimented on in the Krupitzer lab. So this is like archival slices of brains. So I've so far done this for 9 species of rodents, species of marsupials, 2 species of bats, and 4 kind of samples from other orders. And we try to do 3 cases per species. This is still like a very low statistical power, but it's at least something. And you'll notice that for some of the more rare species, like the slender-tailed dunnart, we only have 1 sample. But we are kind of focusing on breadth of species versus depth of 1 species. And I think similar to what was shared in the last lecture about deep learning, like we're kind of seeing a change in paradigm in science where we have so much more data that it's more important to start connecting and collecting this data into a standardized method so we can do analysis on this data to make new discoveries, as opposed to, personally, I think, continuing to deprive animals and experimenting on them. And just a fun example of why I think meta-analysis is cool. There have been more and more discoveries that have been made just based on scrubbing data sets. So my favorite example of this is using this public data set of genomic information, and they were able to get evidence of a relationship between magnetoreception, which is like ability to sense Earth's magnetic fields in a number of different species. And they were able to connect this to a single bacteria that's a symbiont with a certain type of receptor. So they're able to show that it's really this symbiotic bacteria that's helping these species do magnetoreception. So it's pretty cool. And they did this solely by scrubbing genomic data sets and doing correlational analysis. So here's a phylogenetic tree that shows the relationships between the species that I've traced so far. And here's an image of all the species. And I'm showing this to illustrate that we kind of picked species. The Kruvitzer lab has like a ton of really interesting cases of species, but we're picking ones that occupy different environmental niches so that we can ask questions about how the niche and behavioral specializations result in different cortical organizations. So the water shrew is an interesting case. And these are all pretty much interesting cases. And for cases like mice and rats, which are more distributed and not as specialized to a single niche, we try to look at laboratory rats, laboratory mice, and wild rats, wild mice, so that we can make comparisons across niches. Just to like situate how much of the mammal lineage we're covering, it's still a pretty small sample, but this is where primates are and this is where humans are in evolutionary history. We do have the northern tree shrew, and that is probably the closest one to primates that we're looking at. Otherwise, we're mostly looking at rodents and marsupials. So we know that cortical expansions are typically related to unique behavioral specializations, but we're still kind of asking how these unique behavioral specializations are reflected in the overall organization of the cortex. So for example, this is the lab mouse, and you can see it has a much larger S1 somatosensory area compared to the water shrew. And we think this is because lab mice do whisking, and they have these vibrissae, and these vibrissae are, the whiskers are represented in the somatosensory cortex, and they use these whiskers a lot to navigate their environment, so perhaps that's why that's there. But if you look at the auditory cortex, it's much smaller compared to the water shrew, and we think this is because water shrews are actually doing echolocation underwater in order to catch prey, so they have a much more expanded auditory area. You can also compare this with the ghost bat, which we also see a very expanded auditory cortex, and they're also doing echolocation to catch prey and navigate. But interestingly, even though ghost bats have like a genetic propensity to have a expanded auditory cortex, it's also very dependent on developmental learning. So there's recent evidence that shows that mother bats actually train the baby bats to echolocate, which implies that there's this combination of activity-dependent learning and a genetic propensity that results in the cortical phenotypes that we see. So the kind of big picture questions that we're asking about sensory evolution is, are there limits to plasticity? So are there limits to like what you can learn to do? My favorite example of this is that humans can echolocate, and there are a number of blind echolocating experts, but it's harder to, if you're a sighted person, it's harder to learn how to echolocate. So is there some kind of limit to what you're able to learn to sense? How do experience-dependent regions get defined evolutionarily? So how does this underlying genetic propensity to echolocate, for example, get defined by evolution? How might new sensory systems emerge in evolutionary time? And how do sensory perception systems change on an individual level based on environmental experience? So if you play piano every single day, you might have like a piano region in your brain. So that kind of thing. So differences in cortical magnification of body features is both based on evolutionary niche and individual embodiment. So this is the homunculus, which is an illustration of the cortical expansion of the somatosensory and motor areas. So it's illustrating, you know, that the hands are magnified, that there's more of the cortex that represents the hand, if that makes sense. And we can do this for other species as well, where we look at, the somatosensory cortex and where it's expanded. So interestingly, this is an illustration of somatosensory expansion, and this is an image of the real data of the actual somatosensory cortex. So we can look at the red regions in all of these species, and they all represent different body features that are expanded in these different species. So again, to kind of talk about why it's important to connect all this data, even just looking at this image, I don't think anyone has really looked at all these species together. So we can just visualize the data and start to make discoveries. Like just looking at this, I'm sure you all could like already make a discovery just from seeing this. But one important thing that we see is like there are species where the piriform cortex, this gray area, is much more expanded than like, it's like almost the same size as the cortex or bigger than the cortex, for example, in the tenrac. So that implies that they're doing a lot with smell, and we wouldn't otherwise be able to see that. And so why are they doing a lot with smell? Well, if you look at the tenrac's niche, it lives in an arid desert environment and it digs tunnels underground, and there's not many features like in this environment at all. So smell became very important probably. And so it's a very endemic species it's endemic to Madagascar, this arid desert region. Whereas if we look at the water shrew, it's a water hunter, and it's also, importantly, it's a water hunter and it's nocturnal. So it's hunting at night underwater, which implies it doesn't have a lot of visual information coming in. And so that could explain why it became an echolocator with an expanded auditory region. So there's a number of statistical measures that we're hoping to do about these regions and kind of correlate that with niche information. So for example, we can look at the auditory cortex in rodents. And so here you're seeing just the cortex in black and the auditory region in purple. And we can look at how much variability versus homology, which is like conservedness, is there across these species. So how much does this purple region kind of stay the same across species? And that will give us a measure of like how evolutionarily conserved it is versus the variability across species, which gives us a measure of like how much plasticity there is. And we can also look within each rodent species category and again look at the variability versus homology. So how much can this auditory cortex change between individuals of a certain species? And if we do this for other regions, it kind of becomes interesting. So for example, this is the frontal myelinated field in orange here. And if we just look at this field, which is just, it's a darker staining region in the front of the cortex. we can notice that it's across species, and we see it consistently across all species. And this is interesting because in humans, this frontal myelinated region is the orbital frontal cortex, and it's believed to be used or useful in terms of decision making. So decision making processes seem to happen there. And so then we can look back at all these other species and say, okay, what's the evolution of decision making? Like, how are all these other species doing decision making? So it's a question of when we see a homology, like if we see that the frontal myelinated field is a homology, we can make inferences about the function of that area. So the other thing we're doing is a neural net experiment, and I'm really glad you explained the convolutional neural nets because I don't want to like explain this too much, but we're doing a pretty simple feed-forward multi-layer perceptron, and we're training it on, and this is with Stuart Wilson of the Self-Organization Lab, and we're training this on the cortical images. So we take these images that are traced, and we have an XY location and a color that's associated with that XY location, and that's what we feed into this neural net. And we do this for a number of different species and connect these into one neural net. And the idea here is that the number of nodes it will take to represent all the decision boundaries to produce the cortical field would be like a measure of complexity of like how much complexity is there in each evolutionary change step, I guess. So right now we can pretty close to accurately like generate these cortical fields that have been trained on images. And then we're hoping to connect this with niche information so that we can predict changes in cortical organization based on niches. So as I said, we're connecting multiple different species into one neural net. So we can generate a cortical field that is across two species, and that would be like the hypothetical common ancestor between these two species. And then interestingly, we can ask about hypothetical sort of future niches. Like we know that a lot of habitats are changing. There's a lot of changes in niches. So perhaps we can like alter the types of information in a certain niche and predict what kind of cortical organization would result. So future steps is to finalize the results from allometry and other statistical analysis of the fields, possibly add species like primates and monotremes, improve the neural net modeling of the niche information, and make this a database that's publicly available so that other people can play with it and make discoveries in order to connect across different types of data. So we could also connect this potentially with like electrophysiological recordings and with more niche information. So yeah, that's it. So thank you to everyone in the lab, especially Fernando and Chris, and the Self-Organization Lab and all of the funders and stuff.
So it's not so much a question as it is a comment. One, I think this is really fucking cool. I think this is just really, really neat. Two, you mentioned that you might be able to predict future niches, possibly. Could you expand on that?
Future niches?
Yeah, that is so fascinating.
Yeah, I mean, it's pretty science fiction-y because it's like, what is a future niche? But I mean, we know that there is environmental degradation. We know that like habitats are changing. But even like in your example where it's like you have these structures that are outdoor, like how does that change compared to like trees? There's like probably more complexity, there's more visual information, maybe there's different temperatures, different sounds. So I think there's two ways to think about future niches. One is like features, like, okay, this area which used to have a lot of water is becoming a desert. So how will this creature who has an expanded representation of like smell, how will it have to change when it's now in a desert or something. The other way to think about it is in terms of like sensory information. So if an area that was very like liquid, like became desert, smells are now different. So we could model this in terms of like smell and say like, oh, it has 100% variety of smell, and now it has like 3% variety of smell. So how does the incoming sensory information from the niche change what the underlying like evolutionary representation of the field is going to be? If that makes sense.
It does. Wow, that's so fascinating. Thank you.
Well, thanks.
Oh, this is a super cool talk. I guess in relation to that same question, has there been like studies on like you take one species that's particular in one niche or one habitat and like clearly in a way that's not threatening to an environment or ecosystem currently there, like transfer that, or like has there been studies on seeing like, you know, people have like domestic pets or like illegal pets and then they release them to the wild and then they become invasive species to new habitats? Like have there been studies like in relation to that of how like those topical organization systems change because of those?
Yeah, that's a really good question. I don't know of many like cortical region studies on that. I know there's lots of research on like what happens when you release an animal into the wild and like what situations will it be able to cope with the release versus like not be able to. And I think that's a really interesting question. Yeah, but I haven't seen much of like the cortical expansion. Yeah.
Yeah, and then just follow up because I'm not really familiar with this. Like when you take like say like you look at the brain at like early birth and you do like the same staining, is it like the same kind of organizational structure even before like exposures to the niches?
That's a great question. And I think there are some papers in the Kruppitzer lab that look at that where it's like, for example, they enucleate monodelphis, which means they cut off visual information, and they do studies like before, you know, like at a developmental time, early on and later on in both blind and sighted monodelphis. So you do see a lot of changes like from development to adult phenotype. But yeah, it's this kind of interplay between like, you know, the visual field is still there in the blinded monodelphis. So there still is a visual cortex, but it's different. Like it's organized, it's still in the same location, but the shape might be different, for example. And we know this with blind humans as well. So like there still is a visual cortex, which implies that it's very genetically specified, like where the regions are, but that region is being used for things like echolocation. So there's plasticity in how that field gets used, I guess.
Okay, yeah.
Yeah.
Thanks.
Yeah, great questions.
All right, thanks. Great job.